There are people on this planet who don't need sleep. Not function on 4 hours people. People clinically studied, monitored in sleep labs, confirmed to sleep 1 to 2 hours a night with zero cognitive impairment and zero explanation science has fully worked out. There are people who can smell Parkinson's before any clinical symptoms appear. People immune to HIV despite repeated exposure. People whose bodies reject cancer spontaneously, without treatment. None of these are folklore. All documented, studied, partially replicated in controlled conditions. The honest position from researchers is the same across the board. The phenomenon is real, we have partial theories, we cannot fully explain it. These aren't gaps at the fringes. Some are sitting right in the middle of fields we thought we understood. Buckle up, because the conglomerate of all human knowledge has some genuinely embarrassing blind spots, and they're walking around in human bodies right now. Lesson 1. The people who don't sleep. In 2009, UC San Francisco researchers identified a mutation in DEC2 in a mother and daughter who naturally slept around 6 hours and woke up refreshed. Follow-up work found further mutations in ADRB1 and NPSR1 in people sleeping as little as 4 hours with no measurable cognitive deficit. DEC2 appears to regulate the depth and efficiency of sleep cycles. People with the mutation extract the same restorative value from fewer hours because each cycle is denser. But why those mutations produce that outcome in sleep architecture isn't fully mapped. Critically, attempts to pharmacologically replicate the effect have not worked. You can't just target DEC2 and get the same result, suggesting the mutation does something more complex than flipping a single switch. The deeper unsolved question is what sleep is actually for, still genuinely debated. Waste clearance via the glymphatic system, memory consolidation, immune regulation, synaptic pruning. Sleep seems to do all of these simultaneously. The fact that some people do it in half the time without apparent consequence either means they're getting away with something the rest of us can't, or paying a cost we haven't figured out how to measure yet. Lesson 2. The woman who can smell Parkinson's. Joy Milne is a retired nurse from Scotland who told her doctors that her husband smelled different, a faint, musky, slightly altered scent, about a decade before he was diagnosed with Parkinson's disease. Her doctors didn't believe her. Then they discovered she had the same prior observation about other people in her life who were later diagnosed. In 2015, Edinburgh researchers gave Joy 12 t-shirts, 6 from Parkinson's patients, 6 from healthy controls. She got 11 of 12 correct. The one she missed, a control she flagged as smelling like Parkinson's, was diagnosed 8 months later. The mechanism is now partially understood. Parkinson's alters sebum production, the oily substance from skin glands, changing the volatile organic compound profile on the skin's surface. Researchers identified specific compounds that differ between Parkinson's patients and healthy individuals, detectable by mass spectrometry. Joy Milne's olfactory system is sensitive to these in a way normal human olfaction is not. What makes this scientifically strange is that it implies a diagnostic capability sitting latent in human biology that medicine had no framework for and didn't know to look for. The adorable little guys in her olfactory bulb are doing something the rest of human noses apparently can't, and we don't know what's structurally different about hers. Lesson 3. Natural HIV immunity. Approximately 1% of people of Northern European descent carry 2 copies of a mutation called CCR5 delta 32, and those people are functionally immune to the most common strain of HIV. CCR5 is a co-receptor on the surface of immune cells that HIV uses as an entry point, like a doorknob the virus grabs to get inside. The delta 32 mutation deletes a section of the gene, producing a malformed receptor that HIV can't grip. No grip, no entry, no infection. This is well understood mechanistically. What's less understood is a separate group called elite controllers, roughly 0.5% of HIV-positive individuals, who carry the virus but maintain undetectable viral loads and near normal CD4 cell counts without antiretroviral therapy indefinitely. They're infected. The virus is replicating, but their immune system is suppressing it so effectively that clinical disease never develops. The mechanism involves unusually powerful CD8 T cell responses and specific HLA gene variants affecting how the immune system recognizes viral proteins. But not all people with those HLA variants become elite controllers. There are additional factors, possibly epigenetic, possibly involving gut microbiome composition, that science hasn't isolated. The 260-pound bruiser of their immune system is running a defense nobody has fully reverse engineered. Lesson 4. Spontaneous cancer regression. There are documented cases, rare but real, of cancers disappearing without treatment. Not remission after partial therapy, disappearing. Spontaneous regression has been reported in melanoma, renal cell carcinoma, neuroblastoma, and certain lymphomas. The leading hypothesis is immune-mediated. The immune system mounts a sudden massive attack on tumor cells, possibly triggered by a concurrent infection or fever. Regression rates in neuroblastoma are higher in children, whose immune systems are more aggressive, and some cases followed severe bacterial infections. The limbic system of immune response, provoked hard enough by something else, sometimes appears to notice the tumor it had been ignoring. But this is hypothesis, not mechanism. We cannot reproduce it, cannot predict who it will happen to, cannot identify what triggers it. Oncologists have studied spontaneous regression for over a century. The phenomenon hasn't gotten easier to explain. Some bodies cure cancer. We don't know how to make that happen on purpose. The scientific pivot, the part nobody can answer. The honest through line. In each case, science confirmed the phenomenon is real, identified a partial mechanism, then hit a wall where the full explanation requires understanding something about individual biology our current tools can't capture. For short sleepers, we can't explain why the mutations produce efficiency without apparent cost. For Joy Milne, we can identify the chemicals she detects, but not why her olfactory system responds when others don't. For elite controllers, we can name the immune features, but not why only some people with those features control the virus. For cancer regression, we can hypothesize immune activation, but can't predict or reproduce it. Biology produces outcomes that fall outside our models. The outcome is real. The full mechanism is a philosophical path unknown. Which means the models themselves are still incomplete. Human biology contains capabilities we're only beginning to catalog, let alone understand. The exit fee. So, some people run on 2 hours of sleep, and we can't replicate it. One woman's nose detected a disease a decade before medicine could. 1% of a population can't catch the most studied virus of the last 50 years, and some bodies eliminate cancer without being asked. The gap between this is real and this is explained is in each case embarrassingly wide. And in each case, the people carrying these traits didn't know they were exceptional. Their biology was just quietly doing things that broke the standard model. You made it to the end, which means the exit fee is due. 1 subscribe, that's the toll. I'm flipping this tablet around like I'm expecting a tip, and you're paying it in the form of a like. Don't, and I'm assuming your MHC molecules are incompatible with mine. And given that you might be a natural elite controller and I'd never know, that's probably the safest assumption anyway. Stay wrinkly, apes. See ya.